I am Dr. B. DEIVASIGAMANI, working as an Associate Professor, CS in Marine Biology Department, Faculty of Marine Sciences, Annamalai University, Parangipettai. Today we are going to talk about principles and methods of vaccine production and fish immunization. You know the fish immunization, lot of techniques is there, particularly how you can immunizations, what are the techniques. So based on that, we are going to develop the vaccine. First, what is the meaning of the vaccination? So the concept of vaccination. The vaccination is a immune response induction. The vaccine, preparation of antigens from pathogens and rendered non-pathogenic and stimulates immune system of the animals to increase the resistance to the disease on natural encounter with the pathogens. The vaccine means ideal vaccines are only which is inexpensive to produce, easy to administer, safe and effective. The role of the vaccine is primary administrations and sensitize the host and build up a protective immune response by way of memory cells. In natural encounters with the pathogens act as a booster. You know the type of vaccines, majorly two types of vaccine is there, the killed vaccines and attenuated vaccines. The killed vaccines, so many things is there, we are going to use the chemicals and physicals method and heat killing type. So these are the types of the killed vaccine. Chemicals basically we are going to use for the formalin, chloroform and the phenol. In physical method and disruption by sonication lysis with the sodium hydroxide, that is one of the physical method, or the detergent sodium sulfate also used for experimental purpose. This is one of the killed vaccine. And heat killing denatures proteins and oxidizes lipids and destroy crucial antigenic sites present on the pathogens. So these are the three types of the killed vaccine. And attenuated vaccine, so deliver protective antigens to the appropriate site and in the proper context to generate correct immune response. You know the two types of immune response is there. One is natural immunity or immune response. Another one is adaptive immune response. So you know that in attenuated vaccine, maybe you are going to correctly generate what type of response, you know that whether it is innate or acute immune response. So not normally use the fish due to the scare of reversions. And the pathogen virulent in the target species may cause infection in unrelated species and cause widespread infections. And what is the recent type of vaccine? You know very well about that. So recently the vaccine development, so many techniques is there. They are going to use some latest techniques in biotechnology, especially for molecular biology, in subunit vaccines and synthetic peptides and DNA vaccine and mutant viruses. So these are the important recent techniques they are going to use the vaccine development. And characteristics of vaccine. You know the each vaccine you have the characters, individual characters and properties. What is the things is there? The safety, that is most important characters. Whether it is vaccine you can have, the safety is a first priority. Whether that vaccine is going to solve the problem, it cure the problem. That problem is disease, they are going to solve it or destroy it, or whether it is disappearing, whether it is whatever it is, but you should have some safety purpose. That is an important thing. That is one of the character of the vaccine. And immunogenicity, you know that the immunogenicity, it is a first you should have the vaccine development, incapable to have 100% result. Maybe you try to give the 100% result. Whether that is one of the important character of a vaccine development. That is one of the properties of the character of the vaccines, immunogenicity. Maybe very good example for the proteins is a very good source for the immunogenicity properties kind of vaccine. And stimulation of the protective immunity and efficacy of vaccine, then specificity. So that also very important character of vaccine. And route of administrations of the vaccine in case of fishes. Why you should have fish? Fish is a animal model. It's one of the safest and easily available species. You cannot go for in mice and rat and human. That is have you can ethical committee. You can go for that in higher animals. Now you should have some proper ethical community. In here in zebrafish also nowadays you are going to use the ethical. Then only you can have for the any experimental and laboratory aspect experiment things. In case of injections, that is one of the route of the administrations. You can have the vaccines and immersion type and hyperosmotic infiltrations and bathing type and spray, that also one of the administration type route of the administration, and oral models. So these are the oral modes. So that is also is one of the route of administration of vaccine in case of fishes. And next RPS, this is one of the important thing. You can have the relative percent survival. A measure of potency of the vaccine and has to be checked invariably for all the vaccine to be marketed. A minimum 25 fish should be used for the experimentations. So what is we can going to calculate it now? That percentage of mortality of vaccinated fish and percentage divided by percentage mortality of control into 100. So this is the percentage survival of things in vaccine. And steps involved in the development of this vaccine, you should know the first identification of disease problems and the potential vaccine. So this is one of the first steps you are going to develop the vaccine. And in scientific investigation, what is that? Disease pathology. Whether it is your systems have a disease or not, whether it is any type of disease, whether it is bacterial or viral or fungal or whatever it is. So that is one of the things, scientific investigation, that is must one. And iatrologies, that also is an important role for that. You can identify the steps, whether it is the vaccine you are going to develop, is it suitable or not. And laboratory challenge system, then choice of antigen, this is very important. You can choose or choice, whether it is antigen, maybe different type of antigens. You know the antigens, protein type is there, carbohydrate type antigen is there, and lipid type of antigen is there. First you are going to choose or choice what type of vaccine you are going to prepare for your experimental study. For example, you know all of very well know that COVID. So COVID is one of the dangerous disease. How you are going to cure it? It is not possible within the days or within the period. It's a long period. You should have first identify the what kind of pathogen going to cause the COVID. COVID is, you no doubt, it's a viral disease. So that viral disease, how you are going to make it to develop the vaccine against the COVID. It's a lengthy process. First you should choose the best antigens. That antigen probably for protein is well and good. Protein type of antigens. So comparatively carbohydrate and lipids, this protein is a good one choice for the antigens. And the vaccine formulation and commercial consideration. So these are the steps involved in the development of the fish vaccine. And not only that, and production and testing, that also important things. How you are going to test the vaccine in laboratory testing, laboratory things, maybe whether it is going in laboratory condition, in laboratory temperature, your vaccine going to work or not. That's also is one of the production test. And initial field testing, whether it is going to have what kind of field, whether it is minus 20 degree field, whether it is a room temperature or whatever it, you are going to have protection for testing. The initial field testing also important for your steps to develop the vaccine. And finally, product licensing. You can all are you develop the vaccine, and finally marketing. How you are going to marketing your vaccine? You get license, then only your product going to sell. And before that you can have the vaccine license. So that way you can have so many procedures there, and step by step you know very well about that freedom from abnormal toxicity and sterility and safety and potency. So these are the criteria how you are going to get the licensing now. But you should your vaccine is a sterilities, safety, that also very important. Potency, your vaccine maybe you are giving within the 2 days or 1 week or 1 month, your problems going to solve now, that is a meaningful, it is a good vaccine. So that way it is potency also important things for you getting easily license. And RPS should be more than 70% and 10 days observation period. So these are the good things you get the license. And stability, how long, whether you are going to have the vaccine, maybe polio, you know very well about the polio vaccine, is a lifelong. Once you can put the polio vaccine a lifelong, you never expecting for the polio disease and other thing. The same in COVID-19 also, you inject now, how many long you can protect your system. Once again you cannot affect the COVID-19 disease. So that is the very big question mark, maybe 2 years or 3 years, after 3 years, maybe that is also stability of your vaccine. So that is most important, you should getting the licensing. And presentation, that is most important. You should present your drugs or vaccines, and after you should going to sell the services. So these are the criteria. So what are the steps involved in vaccine development? I would say this is not for entire things, this is some small idea how you are going to develop the vaccine in case of fishes, especially for aquaculture and the fishery people. They know very well about how to develop the vaccine in aquacultures, for especially fish and shrimp cultures. Thank you very much for the opportunity.